is, are we talking about in condition C or in setup C?
Okay. Still air. For still air, we know either way wind, either moving or uh, still, it has some impact on transpiration. But now, just give me
Yes.
a second. Patie dakika tu moja.
Okay, thank you for that input. So when we look at these setups, we can start from setup A. There is still air, of course, that is wind, that is still, that is not moving, meaning wind at low intensity. Then also in sunlight and shade. So there is that part of sunlight, and also there is that part of shade. So something could be in sunlight, but if it is shaded, then it means not sufficient light can reach it. Then in B, setup B, we have moving air only, and that is purely wind that is moving. So it means it is a wind of relatively high intensity. Then in setup C, we talk about still air and bright sunlight. So there is that still air, there is wind that is still, and of course there is bright sunlight. So there is wind and sunlight intensity in that setup. And in setup D, we talk about still air. So there is no moving wind, and of course in a dark chamber, so there is no light. So in setup C, maybe if I give you the response, now that I have outlined what conditions are in those various setups, we can say that in setup C, there is a high rate of transpiration. And this can only be attributed to sunlight, because when sunlight intensity is increased, therefore it means that the stomata will open fully, because guard cells, they contain chloroplast, which has chlorophyll to trap sunlight. Therefore, photosynthesis occurs, opening the guard cells fully, and thus the stomata is always open. Therefore, we have high rates of transpiration. So that takes us to Roman 2, condition D. So why is it that in condition D, we have less rate of transpiration of 0.20? So who would help us account for that? Any response from the group? Any response from the group? Yes, I can see Melvin. Melvin, you can unmute and speak.
In setup B, there's a low rate of transpiration due to the fact that it is kept in a dark chamber, hence there is low light intensity, meaning that the stomata won't open fully, hence the low rate of transpiration.
That is very good, Melvin. So having talked about condition C, condition D is exactly the opposite of what we have in setup D. So in the dark, there is no light, meaning at night, always what we learn in photosynthetic theories is that if there is no light, then the stomata will close. Hence, there will be low rate of transpiration. So that makes those 3 marks, 6 marks of that question. Part B, name the apparatus used to measure the rate of transpiration. This one you can indicate in the chat. What is the name of the apparatus used to measure the rate of transpiration? You can indicate that in the chat so that I can see. Let's indicate that in the chat so that I can see. I see the chat is clear. No one is speaking anything. Please let us indicate that in the chat. Some people would rather say it. So we have Charles Karimi. Charles, kindly unmute and speak. We are waiting for you, Charles. Kindly unmute and speak. Yes, I can see someone writing his name as potometer. That is it. Mr. Potometer. That is the instrument that we are talking about. Maybe you can unmute and also say something. Oh, he is gone again. It's okay. So that is that instrument or that apparatus is called as a potometer. And I do believe we need also again to understand the precautions that we need to take while using that apparatus. So which precautions are we supposed to take when using an potometer? Can I see responses for that? Yes, I can see Charles has raised up his hand. Charles, you can unmute and speak. What are some of the precautions that we need to take when using a potometer? Again, Charles has lowered down his hand. Oh, okay, I can see from our chat, we were talking about the potometer. That is very, very okay. So what are the precautions that we need to take while using this apparatus? You can either raise your hand or you can put it there in the chat so that I can also see. There are so many precautions. Yes, we have Kipchumba. Kipchumba, you can unmute and speak. The precautions that we need to take while using a potometer.
One of the precautions that we should take is that the leafy shoot should be cut under the water to maintain transpiration stream.
So the leafy shoot should be cut underwater to prevent blocking of the xylem. That should be it, to prevent blocking of the xylem. Any other precaution that we need to take while using this apparatus? That was good, thank you. Any other? So maybe we can give other people. I can see Ian Ruto also has carried up his end. Ian, kindly unmute and speak.
The oil should be applied to prevent evaporation of water from the beaker.
So where is that oil supposed to be applied?
There at the top of the beaker, at the surface of the water.
So we can apply Vaseline, and that Vaseline is always applied at the rubber tubing where we have our leafy shoot inserted. So that is where we need to apply it. I can see also in the chat, from the chat, I can also see other messages. Nothing is in there in the chat that is streaming that is new. So apart from that, we talk about keeping the system airtight, and keeping it airtight, as I can see someone saying here, is done by application of that Vaseline. Then we also need to ensure that there are no air bubbles in the setup at the beginning of that experiment. Someone called keeping it airtight needed to speak. He has gone again, no problem. So those are some of the precautions that we need to take when using a potometer. Part C. Give one modification in the stomata of xerophytes that reduces the rate of transpiration. Give one modification in the stomata of xerophytes that reduce the rate of transpiration. So can I get responses for that? Can put them in the chat, or yes, someone called small stomata, you can talk. You can unmute yourself and speak. He's gone again. Yes. APPS, you can unmute and speak.
Have reduced aperture to prevent excessive loss of water.
Yes, that is one. Any other?
They have reverse mechanism. That is, they open during maybe the night and close during the day.
That is not really. I know that is a phenomenon, but it's not a structural modification. So when you talk about reduced tomato aperture, that is right. But the other one, would not easily score here. Let us also hear from others. Yes, Ken, you can unmute and speak.
They have sunken stomata.
That is very good. They have sunken stomata. Any other apart from sunken stomata? Fewer number of stomata, not really. There is one other that is remaining. Fewer number of stomata, not really. Yes, Ken, you can unmute and speak again. Ken,
No,
you can
I'm
unmute.
done. I'm done. I don't have any other. Sorry.
Yes, there is one that is remaining. Remember, guys, here we are talking about the modifications. Melvin, you can speak.
Hairy stomata.
Hairy stomata. And those are the three structural modifications that are there in the stomata that brings about stomata of xerophytes that brings about a reduced rate of transpiration. Let us remember, I know stomata is one of the structural factors that affects the rate of transpiration. It has several things in it. We talk about midday closure, we talk about reverse stomatorhythm, and also the ones that we've just talked about here, being sunken, being hairy, and small stomatal apertures. When we look at all these, there are only three that are structural modification. Ile ukarabati imefanyika or ama imefanyiwa, the structure of that stomata to bring about reduced rate of transpiration. So we talk about sunken stomata, hairy stomata, and small stomatal apertures. And that makes the three of them. So any of those three can score, while others are phenomena, but not structural adaptations. Question number 2. Sickle cell anemia is a disease in which people produce abnormal hemoglobin in their red blood cells. Letter capital H represents the gene for normal hemoglobin, while letter capital S represents the gene for abnormal hemoglobin. Heterozygous individuals are said to have sickle cell trait. Question A. If both parents have sickle cell trait, work out the proportion of their offsprings that have sickle cell anemia. So this is 5 marks and you are told to work out. So you need to show you are working. And because we are not restricted, we can either use a Punnett square or we can use a genetic cross. So can we do that cross and maybe in 2 minutes give us the proportion, and the proportion we talk about ratios of their offsprings having a sickle cell anemia. Can we do that and get the responses right now? So what is the proportion? Can we have responses? I see some people have done it already. Yes, let me see from our chat. Our chat still there is I don't see anything from our chat, but I can see Kipchumba has raised up his hand. Please unmute and speak.
Mr. Harman, before you go to the next question, I'd like to ask for some clarification on the previous question.
Yes, please.
In the previous question, the table, we had 3 things: the conditions, the relative humidity, and the transpiration.
Yes.
what maybe the relevance of the column of relative humidity? Because in answering the questions, we did not consider it in any way.
So that is a very good question. When we look at setup C, there is bright sunlight for the condition. So the condition in C, we are talking about sunlight and how it influences the rate of transpiration. It is a very good input again that we talk about relative humidity, just like you have said. So again, when we talk about relative humidity, that is the amount of water vapor that is held by the atmosphere at a given point. So again, if the humidity is low, we can compare the saturation deficit. That is to say, we compare what is inside the leaf and what is outside the leaf. So if the outside of the leaf is having lower humidity, then of course we will be having a higher rate of transpiration. That is why in setup C, again, we can also attribute that to having a high rate of transpiration of 3.75. Then again, when we come back to setup D, because the atmosphere is saturated again, and we are not told if this is out of 100 or should be out of 100. So it's saturated with the water vapor. That will help us again explain the reason why we have low rates of transpiration, because the atmosphere is saturated with vapor such that the one in the leaf will have nowhere else to escape to. But again, if you look at it critically again, the conditions in column A will be of a very high input again when it comes to opening and closing of the stomata, as we discussed before. So that is also a good perspective of looking at it in giving us explanations on why it is possible that the rate of transpiration is lowered. But I do believe that column 1 contains a very bigger portion of what we are trying again to discuss here. So thank you for that input. And I agree with you that relative humidity also contributes to water loss. Thank you.
Thank you.
So let us move to number 2. So for those who have done number 2, maybe let us get also your input. We were talking about parents that are having sickle cell trait, meaning parents that are heterozygous for the trait. And we were told to work out the proportion of the offspring that will be having sickle cell anemia. So who amongst us has done that cross, and maybe they have gotten the response? Yes. Chat is disabled. You raised up your hand. I don't know why the chat has been disabled. No problem. We can just stick to speaking here. Yes, chat is disabled. You can. He is disappeared again. Who has gotten that proportion so that we can see the answers that you've got? Yes, we have Nikki. Please, guys, don't raise up your hand and then you disappear again. Yes, we have Girisham. Girisham, you can unmute and speak. Girisham, you can unmute and speak. Yes, Girisham, we are waiting for you. What is the probability? Work out the proportion of the offspring that have sickle cell anemia. That should be a fraction. What proportion is that? So I can see someone here is saying HH capital, HS capital, HS, and then we have HS. Maybe you can unmute and speak. Please unmute. This guy who has written genotypes on his timeline, kindly unmute and speak. Yes, I can see someone called 25. 25, you can unmute and speak. I can see Victor Kibe. Victor, you can unmute and speak.
Yes.
So tell us what you've done to get your fraction or your proportion. Yes.
So I use the parental phenotype of sickle trait man and sickle trait woman. The parental genotypes are HBA, HBS, and HBA, HBS. So, by use of a Punnett square across the two and found the first genotype as one HBA, HBA is to 2 HBA, HBS is to HBS, HBS. And the sickle cell, the one who has sickle cell anemia is one child. So, and there are 4 of them. So, it's the 1 out of 4, which is 25%.
That is very, very good, Victor. Thank you for that. So if the parents are having the trait, it means that the parents are heterozygous, so that we have their genotypes to be HBA, HBS.
Yes.
So if you do that cross between parents that are both heterozygous, you'll get a ratio of 1:2:1.
Yes.
Whereby you will be having only one child who will be having sickle cell anemia. So it will be 1 out of 4, that is 25%, or a quarter of the offsprings. And that will give you 5 marks. Please let us remember that we show the sign of fusion in that cross, the sign of fusion of male and female genotypes, not between the gametes. I always say ni parents ndio huwa wanapandana. That is where we get that cross. And I'm so sorry to say that, but that is it. The cross is between the parental genotypes and not the gametes. Part B, explain why sickle cell trait is more prevalent in tropical countries than temperate countries. Explain why sickle cell trait is more prevalent in tropical countries than temperate countries. So can we have responses for that? Yes, I can see some people are so alert here. Some people are so alert here and they want to give us answers. Yes, Mark. Mark, you can unmute and speak.
Excuse me, teacher. I have a question
Yes.
on part A.
Yes, please.
On part A.
Yes.
The genotypes will be HB, H capital, and HBS.
Which is also capital.
Okay, thank you.
The reason why we are saying it is capital, and I'll explain that to you. These alleles show codominance, in that if someone is having an heterozygous, if someone is having heterozygous alleles, that is both H and S, then this person is going to have half of their red blood cells are normal and half of them will be sickle cell trait, meaning both of the alleles will express themselves phenotypically, because we are going to have a sickle-shaped red blood cell and also a biconcave red blood cell. So it is a codominant. And please let us note that this is not sex-linked. This is not sex-linked. The HB Victor placed there is just for an abbreviation for hemoglobin. You can as well do it without that HB. You can use HS or HH or SS. Thank you for that. I hope you answered. So for part B, explain why sickle cell trait is more prevalent in tropical countries than in temperate countries, and we want to hear responses. Not the same hands all the time. Where are others? Are others sleeping? Yes, Alan, you can unmute yourself and speak.
Yes, it is a trait that the people in the tropical countries developed to deal with malaria.
Just like that. Any other further explanation you can give us to convince us that whatever you've said is true?
Yes. Since malaria was more prevalent in the tropical countries, natural selection took place and the blood cells developed a sickle shape to deal with the Plasmodium parasite.
That is very good. So at least you are giving us the difference between the tropical country and the temperate country. So malaria is most prevalent in the tropics, and as a result of natural selection, there are mutations that occurred bringing about those sickle cell traits because it confers the resistance to malaria or that malarial parasite, Plasmodium, which cannot survive in a sickle-shaped red blood cell. Part C. Name any other disease caused by gene mutation. So other than sickle cell trait, what is the other disease caused by gene mutation? Yes, I can see Sami. Sami, you can unmute and speak. Seems he doesn't want to unmute. Yes, I can see someone written here, hemophilia. So we have hemophilia as another example of a disease caused by gene mutation. I think we can proceed to question number 3. Equal volumes of 3 different sugar solutions were placed in whisking tubing X, Y, and Z. The tubings were placed in a beaker containing 5% sugar solution. The setup was left for 2 hours. The results were as shown in the diagram below. So in the first diagram, we have at the beginning of the experiment. Then in the second diagram, we have at the end of the experiment. So for part A, it is asking, name the process being investigated. Part B, what is the significance of setup X in the experiment? So let us answer those first two. The process being investigated and the significance of setup X. So we have APPS.
Excuse me, teacher.
Yes, please.
Sorry, but I have to bring you back to number 2. Number 2A.
No problem. No problem, yes.
I was asking if we are supposed to use the letters provided. That is H of
Yes.
S and S only.
Yes.
Oh, thanks.
We were only supposed to use letters provided. In fact, what Kibe gave us, that is HBA and HBS. That would score wrongly, but the explanation was right. It should be H and S only. So kama anatumia HB, that is the abbreviation of hemoglobin, anafaa kuwa anaweka HB, H juu, then HB, S juu, to show that letter H is for normal hemoglobin and letter S is for the abnormal hemoglobin.
Oh, thank you.
You are most welcome. And by the way, if when we look at genetic cross, the genetic process that we have, I hope you can still hear me. All right, sorry for that interruption. I was saying that when it comes to genetic crosses, there are about only 5 known diseases that are sex-linked. So don't try and link any other disease to the sex chromosome in human beings unless otherwise stated. Those diseases for Y chromosome are premature baldness and tuft of hair in the pinna and nose. For Y chromosome, sorry, X chromosome now, we have color blindness, hemophilia, and Duchenne muscle dystrophy, and those are linked only to the X chromosome. So don't link any other kind of trait unless stated otherwise. Those are the ones that are known to be linked to sex chromosomes. So don't link sickle cell trait to a sex chromosome. It won't work out for you. Thank you. So let us go to number 3. Name the process being investigated and what is the significance of part X. So let us have responses for that question number 3. Yes, I see someone called Osmosis has raised up their hand, so you can guide us through. You can unmute and speak. He doesn't want to speak. Yes, Sibau, can you unmute and speak? They are going again. Yes, we have Argento. Argento, can you unmute and speak? You can please unmute and speak.
Osmosis.
So that is osmosis. What about the second question, which is asking what is the significance of the setup in the experiment?
I think it serves as a control experiment.
Why would you say so?
Because it's to compare the results of setup Y and Z, ensuring any changes observed.
Yeah, that is very, very okay.
Yes.
So we proceed to part C. Account for the observations in setup Y and Z. Account for the observations in setup Y and Z. So we can see Y and Z.
Yes, yes.
So can you help us again account for Y and Z? So we can start with Y.
Sugar solution in Y is less concentrated than that in beaker solution. Water will move out of the tubing, causing it to shrink.
Which process will involve, which process will be used by water in its movement?
Osmosis.
That part, please don't always forget about it. So while giving that explanation, you talk about it in that order. What about Z?
Sugar solution in Z is more concentrated than that in the beaker. Water will move into the tubing, causing it to expand.
That is very good. Thank you. Thank
Yes,
you.
yes. Okay.
Thank you for that response. It's very good. And maybe I just want us to note the following when it comes to physiological processes. One, when dealing with physiological processes, before we account for anything, first we must always start by outlining or making an observation in the difference of the solutions or concentrations that we are having. For example, in osmosis, we majorly talk about solutions, whereby you can be having the solution or concentration in the cell sap and not in a potato or not in any kind of cube, in the cell sap, and you compare it with the solution that is around that cell sap. In diffusion again, for example, when we come to gaseous exchange, how will we explain that oxygen diffuses into the lenticels? We will simply say or state that oxygen in the atmosphere is at a higher concentration than in the lenticel. First, we outline the difference in concentration, like you have just done. And then, secondly, we mention the process by which, and not through which, by which these particles move. So we have three main states of matter: solid, liquid, and gases. Most gases and liquids diffuse, except water, which moves by osmosis. Then when we come to mineral ions, those move by active transport, majorly, but also diffusion plays a role in movement of mineral ions, depending on the concentration gradient. And lastly, when accounting, you talk about the observation that comes as a process of movement of whatever liquid or whatever substance you are talking about. For example, like we have said, Y is less concentrated, or it is hypotonic to solution in the beaker, that is the sugar solution in the beaker. As a result, it will lose water. And then what is the observation? The level of Y goes down. What about X? In X, we talk about X being hypertonic to the solution in the beaker. That is the first concentration gradient that we're talking about. Then water moves by osmosis into solution Z. And what would be the observation? The volume of Z increases. So there are those three things that we always must outline very well to get us the 3 marks for accounting. In diffusion, for example, when we are looking at a factor size as a factor that affects the rate of diffusion, when we look at size, for instance, when we have starch solution, and then we also have iodine solution. When, for example, iodine moves and gets into the whisking tubing, of course, there will be a color change. And that color change is what is going to cause the observation that we are going to make. So that is how we do the accounting for those various parts. You talk about the difference in concentration. Then you talk about the process by which that substance moves. And then lastly, you talk about the observation that is made after the movement has occurred. So I hope you've noted that down. Part D. Name a structure in cells that can be compared to whisking tubing. That should be very easy. Any response? Name a structure that can be compared to whisking tubing. Yes, we have Melvin. Melvin, you can unmute and speak.
Membrane.
That would be the cell membrane. What about part E? Explain how high temperature, above 40, will affect the process being investigated in the cells of an organism. Melvin, can you tell us that? He has gone MIA. Okay, can you help us explain that? How would temperature above 40 affect the process being investigated in the cells of an organism? Maybe he's not in a position to. Maybe someone else, because I can't hear him speak. Anyone else who can help us explain that? Any hands up? How is temperature affecting physiological processes in organisms? Temperatures above 40. Any response? That should be an easy one. Yes, we have APPS. Can unmute and speak?
Teacher, I'll try.
Yes, please. Your input is highly appreciated. Try.
I believe temperature above 40 degrees denatures the cell. Hence,
Proceed.
I think osmosis will not take place, or the normal functioning of the cell will not take place.
Okay, that is a very good trial. Let us also hear other people's input. That is a good one. Thank you. Yes, we have Ken. Ken, you can unmute and speak. Yes, Ken, you can unmute and speak. He's not in a position to explain that. Can someone else? Yes, there is Joseph Ngugi. Yes, Ngugi Joseph, can you unmute and speak?
I think it will drop drastically or sharply.
Why? Why would you say so?
So due to high temperatures, you say the cell membrane, which is because we are investigating osmosis, the cell membrane will be denatured, making so water will not move through the cell membrane, hence the process will not take place.
Yes, that is a good one. So we say simply that temperatures above 40 will denature proteins. That is to hit the nail on the head. Remember, the cell membrane is made up of a protein layer and two lipid bilayers. So if the temperatures are above 40, they will denature the protein there, and that will interfere with the property of a cell membrane to be selectively permeable. So anything would be allowed to pass across the cell membrane because its functionality of being sensitive to temperature and pH shall have been destroyed. So part number 4, kindly mute yourself. Who is that? Kindly mute. Joseph.
Excuse me, teacher.
Yes, please. Someone wanted to ask a question. Yes, please.
Can you kindly repeat the explanation for that previous question?
Okay, the explanation is temperatures above 40 will denature the proteins, hence interfering with the integrity of the cell membrane. That is how we explain it. So we go to question number 4. The diagrams below shows changes in a life cycle of a flowering plant. Name the part labeled A, B, and G. Name the parts labeled A, B, and G. So we have those various parts, A, B, and G. Can we name those parts? If someone is writing the names here, I would really want you to speak because, again, interacting brings us to talking about so many more things than when you write. Yes, this guy called A polar nuclei, that. Can you please unmute and speak? I see you've written the right answers, but can you kindly unmute and speak? Probably he doesn't want to speak. Let us get someone else who wants to speak. So we can go for Andrew. Andrew has not talked before. So Andrew, how would you label those parts labeled A, B, and G? You can unmute, yes, please.
A is the polar nuclei.
A is polar nuclei, that is good.
B is egg cell.
B is egg cell, that is good.
Then G is tube nucleus.
Then G is tube nucleus. All those are good. What about the cells adjacent to B? How would you call those?
Which ones?
The ones adjacent, just close to B. How do you call them? He has gone MIA again. Let us get someone else to help us. Andrew has kept quiet. So let us get someone else to help us. Yes, we have Sir Charles. Charles, kindly unmute and speak.
Cells adjacent to B are synergids.
What about those towards the North Pole, above A? Charles. Are you there, Charles? He's gone again, MIA. We need to chambua this question first before we answer those other questions. Yes, I can see Ian. Ian, kindly unmute and speak.
The antipodal cells.
Those are the antipodal cells. What are their functions, the antipodal cells? Ian, the ball is still on you. Ian again has gone quiet. We need to talk about this thing better and better. Can we have someone else? We have Melvin. Melvin, you can unmute and speak.
Provide nourishment to the developing embryo.
Provides nourishment to the developing embryo. Really? I doubt so. Well, the ones that are above, that is above A, we call those as antipodal cells. And these three guys, ama hizi mafiga matatu, they always direct the growth of the pollen tube. I hope you remember what we call as chemotropism. This is an example of chemotropism. Growth response, tropic response, in regards to chemical concentration. So that directs the not the synthesis, but the growth of the pollen tube. Then we have a part A, that is the polar nuclei, and part B, of course, that would be the egg cell, and down below are synergids, which nourishes the egg cell. Please, this diagram of the ovum or the egg cell, I want us to note something. It can always be drawn in a manner likely to confuse us. How? Remember, when double fertilization is taking place, there is that point we call as the micropyle. That point through which the tube nucleus enters this egg cell or this embryo sac. That micropyle is always closer to where the egg cell and the synergids are, and not where the antipodal cells are. So now, when you look where the egg cell is down there, there are three. Then again, the same three are on the opposite, and those we call them as antipodal cell. The diagram could be drawn to have the micropyle up where the antipodal cells are. And those now will cease to be antipodal cells, but there will be the egg cell and the synergids. I hope that is taken well. Then, of course, on this other end, we have part G, and that would be the tube nucleus. So state the function of part labeled D. State the function of the part labeled D. So what would be the function of part labeled D? Let us get responses. If someone is talking about transport male gametes to the embryo sac, and that is basically it. But Mark wants to say something. Mark, please say something.
It is used to guide the development of the pollen tube and the generative nucleus down the style.
So it guides. Guiding the growth that is done by the tube nucleus, okay?
Yes.
So when we talk about that tube itself, or the pollen tube itself, it allows for those gametes to be transported all the way to the embryo sac. That would be the very, very right response.
Okay.
Thank you. Then let's proceed. What will part A develop into after fertilization? What will part A develop into after fertilization? So any responses? And part A is the polar nuclei. What will it develop into after fertilization? Any responses? Yes, we have Francis. Francis, you can unmute.
Yeah. Can you say the endosperm, into the endosperm?
A develops into an endosperm after fertilization. We can talk about primary endosperm, okay?
Yeah.
It forms the primary endosperm. Okay, that develops into an endosperm. That is very, very okay. It is the endosperm.
Yeah.
Then what about part B? What will part B develop into, Francis?
The egg cell. For
Yes,
sure, I
what?
don't know. For sure, I
Okay,
don't
no
know.
problem. I know there is someone who will who knows. Let us ask them also. What will part B develop into? I see there is 25 to 43. Kindly unmute and speak.
Okay, I think the answer is an embryo.
Yes, it will develop into an embryo. So maybe again to jog our memories a little. Whatever we have here are showing two gametes in flowering plants. That is the embryo sac and the pollen tube. Of course, the pollen grain that develops into a pollen tube. So it is quite important to note that the two male nuclei that are found in the region.
Najibu swali moja na kuongezea ingine juu.
Kujua kama kuna contents.
Usikimi.
Unajua examiner nakosa
You can mute. We can hear what you are saying. Kindly mute. Yes, so we are saying that part D has three gametes, one labeled G and the other two. Those other two gametes are generative male nuclei, which one fuses with the egg cell B to form a diploid zygote. And that diploid zygote will later develop to form the embryo, which has three parts: plumule, radicle, and the cotyledon. Hence, we have the seed. Then the other male nucleus will fuse with part A to form a triploid endosperm. And when we form the triploid endosperm, that will now develop to form the the endosperm that now stores food for the seed. So that is what we have after fertilization. So I think we can proceed. Define the term parthenocapy. How can we define the term parthenocapy? Or how is the term parthenocapy defined? Yes, any new faces? Can see Charles, Andrew, Ian. So let's go for CMF2. CMF2, you can kindly unmute and speak. CMF2. How do you define the term parthenocapy?
This is the growth of fruits without fertilization taking place.
So we use the term development. It is the development of a fruit without fertilization, or fruit formation without fertilization. We also have another similar term used in animals that refers to development of an organism or an animal without the process of fertilization. How do we refer to that term? So in plants, we call this as parthenocapy. How do we refer to that plants in animals? Yes, we can see Barasa. Barasa, kindly unmute and speak. Baraza, you can unmute and speak. Baraza has gone MIA. Anilov, can you unmute and speak?
Parthenogenesis.
That is called parthenogenesis. And a good example of an organism that carries out that process of pathogenesis is called Drosophila melanogaster, or the fruit fly that we know of. Part D. That was good, thank you. Name 2 features in flowering plants that prevent self-fertilization. Name 2 features in flowering plants that prevent self-fertilization. And that should be an easy one. That should be a very, very easy one to go with. Yes, Mukira, kindly unmute and speak.
Self-sterility.
Self-sterility, any other?
Protandry.
Protandry, any other?
Heterostyly.
Heterostyly. We have heterostyly. So those features that prevent self-pollination and enhances cross-pollination, we have heterostyly, like you've said. Hetero means different, like we have heterozygous, heterotrophism, heterogametic. So when you talk about heterostyly, style means the style. So the style grows longer than all other floral parts. Therefore, it will prevent self-pollination. We have protandry and protogeny. When I see that word GY, sorry, the letters GY in a sentence, like gynoecium, protogeny, those words tells me something. There is these doctors who specialize in female reproductive health. We call them as gynecologists. So that always tells me that whatever I'm trying to talk about deals with the female parts only. So when you talk about protogeny, we simply mean that the female parts of the flower, that is the pistil, it matures first before the male. And protandry would be the opposite. And it is protandry, not protandry. Protandry. And then we have also monoecism and dioecism. Not monoecious and dioecious. Monoecism and dioecism, where mono means one. So there is one plant having male and female flowers in different parts. Mono, one. but different parts of the same plant. Then we have dioecisim, where di means two. Therefore, we have two plants of each either has a male or a female flower. So meaning the male and female flowers are found in two different plants. And of course, you have self-sterility and self-sterile. Something sterile is not something that is not able to fertilize. So if we're talking about self means an individual. So if there is the anthers from the same plant, they cannot fertilize the embryo of that, or they cannot germinate in the stigma of that same plant. And those are the features that we have. Question number 5. And if you have any question, please always try and raise up your hand. Question number 5. The diagram below represents one of the joints in mammalian body. You can see the joint. Name the type of joint shown in the diagram. Name the type of joint shown in the diagram. So what are the responses for that? Name the type of joints found in the diagram. Yes, we have X3G. X3G, you can unmute and speak. What type of joint is this? I can't hear you. Don't know if others can hear you. What type of joint is this? Maybe he has an issue with his network or something. I can see, yes, there are other people coming in. We have Rexvin. Rexvin, can you unmute and speak?
It is ball and socket.
Why would you say that it is a ball and socket joint?
Because you can see part Z is the femur.
So part Z is the femur. Is that a very concrete reason? So for a ball and socket joints, we can see that there is a depression, and the other bone which fits into that depression has a rounded head. And that is how we define a ball and socket joint. There is a ball, a rounded head, and a depression into which that rounded head fits. That is very good. That is a ball and socket joint. And I know someone would talk about a synovial joint. Synovial joints are any joints that are having the synovial fluid. So we have two types of synovial joints. In fact, there are three. Ball and socket joint is a type of a synovial joint, like this one we are having here. At our shoulder region, where we have the head of humerus fitting into the glenoid cavity, that is a ball and socket joint. Also, this one we have femur fitting into the acetabulum of the pelvic girdle. That is also ball and socket. Then we also have hinge joints. Hinge joints are found at the elbow. That is where we have the sigmoid notch and trochlear of the humerus. Again, we have hinge joint at the knee, where we have the condyles of the femur, and we have the smooth facets of tibia. Also, we have hinge joints at our phalanges. And lastly, the other type of synovial joint we have is the gliding joints, the ones that we have at the distal epiphysis of both tibia and ulna, respectively, to articulate with the carpals and tarsals, also respectively. Also, we have gliding joints at our vertebral column, and these joints, they only allow for a small degree of movement. So this type of joint, we need to hit it on the head and say, that this is a ball and socket joint. Reason: one bone has a rounded head and the other feeds into a depression. Let's proceed. Name each of the parts labeled Z and U. So what would be part Z and what would be part U? So any responses for that, part Z and part U? So I can see Rexio. Rexio, you can unmute and speak.
So the parts are femur and articular cartilage.
So part Z is femur, that is very, very right. And part U is?
Articular cartilage.
Articular cartilage, that is very okay. Let's proceed to the next one. State two functions of fluid in W. State the two functions of the fluid in W. So how would we call that fluid and what are its functions? So we can be having other people also giving us responses. We can see Osman here. Osman, you can unmute and speak. Osman, you can unmute. Osman is gone. We have Argento. Argento, you can unmute and speak. What is that fluid and what are its functions?
Teacher Kwanza, it's agent 00 and the fluid is synovial fluid.
I didn't know that was 0.
Yes,
So
yes.
the fluid is
Agent
sigmoid
00.
fluid.
Yes, yes.
It is okay. It's
Okay.
okay. It's okay. So that
And
is the synovial fluid. What are its functions?
number one, it's for lubrication. It reduces friction between cartilaginous surfaces during movement.
So it lubricates and lubrication reduces friction.
Yes.
Yes. Any other function? Because we need another function.
It acts as a shock absorber.
We don't talk about acting.
Yes.
It cautions shock or it absorbs shock.
Yes.
Yes, let us know that in biology we don't have theatrics. We talk about those things doing those functions. Okay?
Okay.
So it is a shock absorber, and lastly we can talk about it nourishes the tissues around that region, the cartilages and the tissues around that region. Apart from this fluid, we again have other fluids in various parts of the body. So we start with the one we find in the meninges of the brain. So in the brain, we have 3 meninges that cover the brain and of course the spinal cord. We have the dura mater to the outside, pia mater to the inside. And we have a cavity called the arachnoid cavity. Which fluid fills the arachnoid cavity? And what are the functions of that fluid? So let us get responses again for that. Yes, I can see Quincy has raised up his hand. So Quincy, you can unmute and speak.
Functions of the. Oh, okay. Please pardon the question.
The question I was asking is,
yes,
we have other fluids in the body that also play crucial roles.
Yes.
And when we talk about the brain or the central nervous system, that is the brain and the spinal cord.
Yes.
They are covered with the meninges. That is the dura mater to the outside.
Yes.
Yes, dura mater to the outside, pia mater to the inside. And there is a space or a cavity in between called the arachnoid, which there is a fluid which fills. Which fluid is this, and what are the functions of that fluid?
Would it be the cerebrospinal fluid?
That is good. What are the functions of the cerebrospinal fluid? Quincy has gone. Let us have Jimmy. Jimmy, can you unmute and speak? Yes, Jimmy, you can unmute and speak. No, Jimmy has also gone MIA. Oh, I can see others are down here. So let's have Nodine. Nodine, you can unmute and speak. What are the functions of the cerebrospinal fluid?
Act as a shock absorber.
I just said about a minute ago that in biology we don't act. Nodine, can you reframe your statement?
Help to absorb shock.
What is the difference between help and act? In biology, you don't help, you don't act. Can you again reframe for the last time, please?
Absorb shock.
That is very good. It absorbs shock or it cushions shock. Okay?
Yes. Yes.
That is one. Any other that you may know of? Any other?
Prevents internal mechanical damages.
Okay, that is a wild, weird trial. Let us also get what others have to say. We have Kipchumba. Kipchumba, you can unmute.
So the other function of the cerebrospinal fluid is it provides nourishment and removes waste products.
So it nourishes the brain and also removes waste products from the brain. Another fluid that we have in the body is the amniotic fluid. And amniotic fluid is found in the amnion. It's secreted by the amnion and found in the amniotic cavity. What are functions of the amniotic fluid? Yes, Ernest. I think I did that wrong. Please have me forgiven. Let us give a chance to Evans. Evans has gone MIA. Let's give this chance to James Ngugi. Yes, James, you can unmute and speak. What are the functions of the amniotic fluid?
It nourishes the fetus.
Nourishes the fetus during the first 4 months of development. Any other?
It keeps the fetus moist.
Prevents dehydration or desiccation. Any other?
Allows for movement of the fetus.
Offers support. It offers support, therefore the embryo can move within the womb. Any other?
It also maintains pressure and temperature.
So it equalizes pressure as well as maintaining or regulation of temperature. Any other? Those are very, very good.
Those, I only have those.
So also it lubricates the birth canal during parturition. And that would be the other one. And let us also remember that in when we are forming the embryo, we have three parts of that. So those 3 parts of the blastocyst that differentiate include one, the chorion, which is on the outside. And of course, the amnion is to the inside, leaving that part we call as the allantois in the middle. The same kind of structure that we've seen in the brain, dura, arachnoid, and then pia. The same which we again also have in the eye. In the eye, we have sclera outside. Then we have chorion. Sorry, yes, we have the choroid, not chorion, choroid in the middle, and the retina to the inner part. So those are again other parts that we need to talk about. Apart from those fluids, again we have the pleural cavity where we have the pleural membrane secreting the pleural fluid, and that is on the surface of the lungs, which also not help, but lubricates the lungs when the lungs are pumping. Then when we get also to the heart, we have the pericardium, which is a double membrane that secretes pericardial fluid. Pericardial fluid, which also lubricates the heart as it pumps blood. I can see other people here have raised up their hand. Do they have questions? Let us see. Andrew, you can unmute and speak. Okay, it seems that they don't have any questions. So I think we can proceed. We are going to part D. Identify the type of muscle found attached to bone Z. Identify the type of muscle found attached to bone Z. The type of muscle found attached to bone Z. So what type of muscle is that? So the same hands every time. Yes, member, you can unmute and speak.
Skeletal muscles.
Skeletal muscles. So apart from skeletal muscles, member, what other types of muscles? Sorry?
Pardon?
That is the skeletal muscle. So apart from skeletal muscle, member, what other types of muscles do we have in the body?
Visceral muscles.
Someone is
muscles.
Oh yes, is member. So we have the smooth muscles and the cardiac muscles.
Yeah.
And these muscles, even from your Malina in the previous exams that you did, you are being asked about them. So at least also you need to note, or at least know, examples of parts in the body where we find these muscles. So like, for example, where in the body do we find the cardiac muscle? Where in the human or in a mammalian body, where do we find the cardiac muscle? Yes, Mark, where do we find the cardiac muscle?
The cardiac muscles is found in the heart.
So heart is made up of the cardiac muscle itself, sindio? Good. Where do we find smooth muscles in the body, Mark?
Smooth muscles are found on the gut and the fallopian tube.
So any passages that we have in our body, those passages, like the ones that you've mentioned, the gut and also in the fallopian tube, they have those smooth muscles to facilitate movement of various things, like food and the ovum, respectively. Where do we find the skeletal muscles?
The skeletal muscles are found on the bones.
So they are found attached to the bones, and it is them that always contract and relax to bring about movement. So again, we need also to note about the differences that we have in these muscles. Like if you talk about the nucleus, if the one is uninucleated, if the other is multinucleated, are there striations or are there not, etc., etc. So those differences, again, you need to outline them well. Then we move to part E. State two differences between the muscle identified in D and those found in the gut. And that is the same question that we have just tried to ask. What is the difference between a skeletal muscle and a smooth muscle? So what are the differences that we have for that? Yes, I can see Alan here. Alan, you can unmute and speak.
Smooth muscles are, well, The muscles found in the gut are smooth muscles, and the differences are that smooth muscles are non-striated, while skeletal muscles are striated.
That is good. Any other?
Smooth muscles are involuntary, while skeletal muscles are voluntary.
Any smooth muscles are involuntary. Okay.
Yes.
And any other?
Smooth muscles are found in walls of internal organs and blood vessels, while skeletal muscles are attached to bones.
That is now a functional difference or what? It does not bring that difference clearly.
Oh. The last one I have is that smooth muscles have a single nucleus, while skeletal muscles have many nuclei.
So smooth muscles are uninucleated while skeletal muscles are multinucleated. I can see
Yes.
someone is asking, why is part U not acetabellum? So again, maybe we go to that diagram and we see. So why is part U not acetabellum? So I would say part U, so we have the synovial membrane enclosing the synovial fluid. And of course, on top of that synovial membrane, we always have a cartilage, a muscular cartilage that prevents shock or friction also. So part U, why is it not acetabellum? So if you have the pelvis here, then this depression should be the acetabellum. But again, I think it's the labeling or the diagram not outlining those parts clearly. We have a cartilage there and also we have acetabellum. So I think because it is not giving out the clear outline, this would be acetabellum or it could be a cartilage because there must be a cartilage there that also prevents friction from occurring. So I think that question of yours is answered. Any other question that might be pending? That was a good one. What about obturator foramen? Thank you. So for obturator foramen, foramens are always holes. And that hole called obturator foramen, it is a hole that is found towards the lower part. You see where the part labeled U is? Yes, towards the ischium. And obturator foramen, that hole allows for passage of blood vessels to other parts of the body, from the legs to other parts of the body. Like the same way we have foramen magnum, that hole that is found in the skull between the two occipital condyles that articulate with atlas at the neck region. So foramens are always holes. And I think you are answered for that, or passages. Any other question that is pending? Any other question that is pending? I can't see any other pending question, but if you have a question, please, you can raise up your hand and ask. And I do believe those people who are asking those questions, your questions are answered well. If you're not satisfied, please carry up your hand again. So we move to part B, and this is not showing our table properly, but part B is majorly graph. So what about graphs should we know when it comes to drawing a graph to score the 7 marks that we have? So this one I'll just speak, and I hope you will be listening. Can someone tell me the time we are supposed to be ending this class? Because I see the numbers are going down. Okay, I can see someone saying Mwalimu time. All right. If time is up, time imeisha. That is very good. Thank you for reminding me. So I didn't know the time we are supposed to end. but I believe the class has been an interactive one. So in our next class, we'll proceed from question 6 to section B. Thank you for listening. Thank you for participating. Have a good day ahead. Bye-bye. That ends our class. Bye.